Thank you very much, Steve. So most of the folks attending today's presentation will already appreciate the fact that multicolor flow cytometry is an increasingly important technique in modern cellular analysis. And if you happen to do multicolor flow cytometry today, you will also appreciate what the graph at the bottom illustrates. This is simply our analysis of the fluorescently labeled antibody catalog of a particular vendor within the industry representative. And this just shows that the typical clone available, a flow cytometry qualified clone, is really only available in 2 or 3 different colors, labeled with 2 or 3 different fluorophores from any one company. And that's true actually across the industry. There are very few antibody clones available in, say, 10, 11, or 12 different colors, many that are only available in 1 or 2. 3 seems to be about the average for the industry. And the problem that this presents for the flow cytometrist doing multicolor flow cytometry is, of course, that if you only have 1 color channel open in your multicolor panel to insert a new antibody into, as you are, say, expanding the panel, if you can't find the antibody you need labeled with the color that fits in that channel, then your choices are really very limited. Either you have to abandon the effort, or you have to engage in a custom synthesis and conjugation project, either yourself or contracting someone else to do it. Either way, makes it very difficult to develop new multicolor flow panel assays with great facility and dispatch. And that's really the challenge that the new technology I'm going to be describing today addresses, and I think addresses optimally. If you think about what the requirements would be for enabling a flow cytometrist to get any antibody needed in any color needed, you really don't want it to be the case that vendors will simply sell all of their antibodies labeled in 22 different fluorophores, because then you, as the end user, would need to have a refrigerator jammed full of little vials of antibodies in order to take advantage of that, or face constant back ordering problems, perhaps. Instead, what you'd like is a just-in-time technology where at the point of use you can say to yourself, I need this antibody labeled with this particular fluorophore. And then you could just mix 2 volumes of antibody and fluor together. They would spontaneously label, and you'd be able to use it at the time you actually need it. Because most flow cytometrists are not chemists, you'd also like it to be a technology that doesn't require any chemistry skills at all. Even as far as a simple separation step at the end of the labeling process, you really don't want to get into that. You want to mix and use technology. If one is going to label small batches of antibody at time of need, the technology to do that necessarily needs to give you perfectly consistent lot-to-lot performance. If you label 1 batch today and another batch 6 months from now, you want the 2 batches to perform absolutely equivalently. Finally, you need it to work in a wide variety of conditions that are common in cell biology research, anywhere from 4 degrees Celsius to 37 degrees Celsius, and in any level of complexity of matrix from PBS all the way to whole blood. You want the technology to work well. As a cocktail and as a single color, you want it to work well, and you don't want any limitations with respect to the types of antibodies it will work with, or with the classes of fluorophores that it will work with. And that's a pretty tall order, and fortunately the technology I'm here to discuss today actually addresses that quite elegantly.